Hello and welcome to another lesson from the A-level series of video lessons from the conscientious biologist Ben Gallagher. This is the second video in the entire A-level playlist, and this is cell ultrastructure, which is a detailed look at cell organelles. There's nothing difficult to understand here, but there is a lot of information about to come at you. But it's really important that you learn everything from this video so that you understand where all the complex processes take place when we move on to higher levels of the course. As always, please do subscribe to my YouTube channel if you haven't done so already, and do follow me on Instagram or Facebook for lots of additional biology information. Thank you. So if you've just come straight from the first video, this is all stuff that you should already know. That eukaryotic cells, which is what we're focusing on in this video, are a cell membrane and a cytoplasm, chromosomes of DNA and ribosomes. If you've got those four things, you can be a compartment that can make proteins, which means you can perform metabolism. All cells, if they're going to be living and metabolically active cells, have to have those four things, no matter what they are, eukaryotic or prokaryotic. Eukaryotic cells, though, and in this case an animal cell, will have a nucleus to protect the DNA, mitochondria for aerobic respiration, and they could have a small temporary vacuole for storage. Plant cells will have a cellulose cell wall around the outside of them to maintain their shape. The membrane will be pressed against that cellulose cell wall so that they can become turgid to have some solid structure for the plant cell. As well as the ordinary stuff, the nucleus, the ribosomes, the mitochondria, they might have a large permanent vacuole, and of course they will have chloroplasts, which are the site of photosynthesis. Now you've got to know all those things for these two cells, but what we're going to do in this video is expand on all of these structures and add a few more. Now all of these little things that are inside a cell performing a job are called organelles. Just like you as an organism have organs, your heart, lungs, liver, etc. Cells have organs of their own, but they're called organelles. So that's what we're going to be exploring in this video. So first up, the most obvious one that you have in eukaryotic cells is the nucleus. Now from GCSE, you would of course know that they contain chromosomes of DNA. You'd know, hopefully, that chromosomes can come in slightly different forms though. And what I've put over there are what's known as single chromosomes, where that's one very long strand of DNA. Now single chromosomes, about midway through a cell's life, will split and copy themselves into two identical pairs which stay together. Those are called double chromosomes, and that's the process of DNA replication. It's a really important part of a cell's life. The reason being, if one cell is going to grow and then split into two, each new cell will need a full set of DNA. So that first original cell has to double all its DNA so that through the process of mitosis, it can share the DNA out and each new cell has a full amount. That's why for the first half of a cell's life you see single chromosomes, and for the second half of a cell's life you see double chromosomes. Of course, during mitosis, those chromosomes actually become visible even under a light microscope. Hopefully you've looked at them through the microscope during GCSE, and then it would be a condensed double chromosome. That should be familiar. If it isn't, please go and have a watch of my video Cells 5 on mitosis. Again, it's GCSE level, but you've got to make sure your foundations are strong, your GCSE foundations, if you're going to do well at A level. Let's get rid of that for now though, and let's focus back on the structure of the nucleus. So you've got these chromosomes. You know the chromosomes are kept in a membrane. That's to give them some protection. But to give them extra protection, it's not just one membrane. You have two membranes, and those membranes hold in the nuclear plasm. So like a cell is full of cytoplasm, the nucleus has a compartment within the compartment that's the cell. The liquid inside there is the nucleoplasm. And you've got these two membranes. So we can say there's an inner membrane and an outer membrane. And the term for that, when you've got two membranes held next to each other, is an envelope. So this is the nuclear envelope. It's called an envelope because think about an envelope that you might put a birthday card in. It's got two layers and you slot your card in between them. Two layers is an envelope. So this is the nuclear envelope made of an inner and outer membrane, and the nuclear envelope holds in the nucleoplasm and the chromosomes float around in the nucleoplasm. Now, you also have throughout the nuclear envelope, you have holes. These holes are called nuclear pores. Now, those are really, really important. Think about what you might remember from GCSE protein synthesis. You need to make a messenger copy of DNA to get out of the nucleus to go to the ribosomes. That's quite a big molecule, so it needs a hole to leave through. The building of that messenger molecule also requires enzymes, which are pretty big, enzymes which were made in the cytoplasm. So you've got to get them back in. So these nuclear pores are basically holes through the membrane that can allow stuff to go in and out while still maintaining the overall protection that the nuclear envelope gives. So it's really, really important you remember about those nuclear pores, and we do talk about them quite a lot when we're looking at cellular processes later on. Another structure that you've got in the nucleus is this. This is called a nucleolus. Now, you probably won't have come across this before. We won't talk about it again in the course, so I'll give you its purpose now. The nucleolus plays a role in ribosome production. It actually makes something called rRNA, which we'll talk about later in the course, and ribosomes are made partially of rRNA. All you need to know for A level is the nucleolus is there, floating around in the nucleoplasm, and that it plays a role in production of ribosomes. So take a screenshot of this, because this is all the information you need to know about the nucleus, which, consider back to GCSE, where all you labelled maybe was the nucleus and maybe DNA. You've now got all those labels that you would be expected to add as well. I'll try and speed up a bit now. So mitochondria, it's the site of aerobic respiration. You know that. If I copy this diagram across from the one that we used previously in the GCSE diagrams, you've got an outer membrane. Now, the fact that I've put outer membrane should be making you think, oh, there must be an inner membrane as well. And there is, but the inner membrane in a mitochondria looks a bit different. It's not like the nuclear one where the two of them run in parallel. The inner one has all these big projections and foldings that push out into the mitochondria. So we generally don't refer to that as a mitochondrial envelope, just because the two layers do very different things. The outer one is smooth, the inner one forms these big projections that move in. And those big projections are called cristae. So within any mitochondria, you will see big projections that stick out into the cell. Those are the cristae. The liquid part that's inside is called the matrix. So inside the cell, you've got cytoplasm. Inside the nucleus, you have nucleoplasm. Inside the mitochondria, you've got the matrix. That's it for the mitochondria. When we do respiration, we'll have to look at that in a lot more detail on what those bits do. But for now, you just need to know them and be able to label them. So thinking about one from a plant cell now, we'll look at a chloroplast, which is the site of photosynthesis. So a chloroplast, it's got its outer membrane, it's also got an inner membrane, and you can see that the outer and inner membranes do follow each other now. So we can call that the chloroplast envelope, and they trap inside the liquid, which is called the stroma. So we've now got lots of different liquids appearing within the compartments within the cell. Okay, stroma and matrix and nucleoplasm, they're all equivalents. They're the liquid trapped within the compartment. Okay, but within the stroma of a chloroplast, you've got these large structures. Now, these are called thylakoids. These are really, really important when it comes to photosynthesis. And again, we look at those in a lot more detail when we do photosynthesis as a process. Now, a stack of thylakoids is known as a grana. Okay, grana can be visible through an through a standard light microscope. The reason why they have their own name when they're just a stack of other things is because they were seen and commented on as, oh, there's grains, and that from the Latin we get grana. So they were first recognized, and then they went, oh, those grains are actually stacks of other things. But the name grana has stuck around. But grana is a stack of thylakoids. Now, also, though, slightly confusingly, given that grana is a stack of thylakoids, within the stroma, you might also find starch grains. Don't confuse starch grain with grana, even though they're both very similar, okay? A starch grain, because starch is made from glucose, and glucose is, of course, made by photosynthesis. So some of the glucose that's made in a chloroplast, made by photosynthesis, polymerizes, forms starch, forms a grain, and can stick around in the stroma. It might be shown on a diagram in the exam, so you might need to label it, so you've got to know everything that's on this screen as well. So take a screenshot of this as well to add to your others. Okay, let's move on to ribosomes now. So ribosomes are essential in protein synthesis. They're the things that stick the amino acids together to build them. Ribosomes, unusually, given most of the organelles, don't have a membrane that binds them together, so they're not membrane bound, but they're very, very small. If I was to put them to scale, there'd be that little pink double dot up there. So let's enlarge that so we can actually see what's going on. And ribosomes are made of two subunits, a large one and a small one. And if you remember from GCSE, the messenger molecule that comes from the DNA, it's like the photocopy of the DNA that can leave through a nuclear pore to go to a ribosome. It gets read there between the two subunits. Now, I'm hoping you remember that from GCSE. If you don't, here's a screenshot from my GCSE lesson on it. If this doesn't look at all familiar, or something similar to this, where you've got this black molecule being the messenger molecule, the ribosome reading it, you've got a carrier molecule bringing amino acids, which are the smiley faces there, so that you can build up that polypeptide. If that doesn't ring a bell at all to you, please go back and watch that lesson. It's this one, Details of Protein Synthesis. It's too important to neglect. If we're still focused on ribosomes though, in eukaryotes, the ribosomes are called ATS ribosomes. Now the S is a measure of size. It's called a Svedberg unit. It's a really, really, really tiny unit of measurement, and I've only ever come across it being used for ribosomes. But just to give you a little bit of detail, in eukaryotes, the ribosomes are ATS, whereas if you look in prokaryotes, they're 70S, which means they're just a tiny bit smaller. Okay, take a screenshot of this if you need to, add that to your notes as well. Okay, so this one I'm not expecting you to have heard of at all. This is endoplasmic reticulum. Now, endoplasmic reticulum, there's loads of it in a cell, and there's a couple of different types, but they all kind of look like this. This is smooth endoplasmic reticulum, and it's basically just a load of membrane. Remember, almost all the organelles are made of membrane, ribosomes being that exception. But this is if we've got some membrane, same type of membrane that the cell is made of, and you've just kind of pulled it and tweaked it and stretched it into a weird shape like this. And all these little stretches and branches are called cisternae. Now, smooth endoplasmic reticulum, you don't need to know much about it for A level. The key kind of things about it is it's involved in the synthesis, storage, and transport of lipids. But membranes are made of lipids. So really what we're talking about with a smooth endoplasmic reticulum is it's basically spare membrane. If you need a bit of extra membrane to make a vacuole, let's say a temporary vacuole in an animal cell, you might pinch a bit of membrane from the smooth endoplasmic reticulum. Or so some might go into the cell membrane so the cell can grow. Okay, but they're essentially just a big load of spare membrane. They do some transport, but don't worry too much about that. The other type is called rough endoplasmic reticulum. Now, looking at the diagram above me, it looks very similar to smooth, but the reason it's called rough is because all over its surface are ribosomes. So if you're looking at it through a light microscope, it looks like it's got a really rough surface. So the rough endoplasmic reticulum is the main site of protein synthesis. This is where a lot of the building of the polypeptides happens. But the rough endoplasmic reticulum is also responsible for transporting those polypeptides. And the way that you can do transport is to use something called vesicles, which are little bubbles of membrane. So we're going to add vesicles to the title there, because they're like this. You can see on the screen, look, they're just little bubbles of membrane that float off from either rough or smooth endoplasmic reticulum. But those little bubbles of membrane that literally just kind of pinch off the end and float away, but they would be carrying something. So in the case of rough endoplasmic reticulum, it's made the polypeptide. It might pull the polypeptide into the cisternae. And just because I haven't labeled it, by the way, cisternae, same on both, it's the long bits. So in the cisternae, you might have the polypeptide, which then might just go to the end, pinch off, and that's a vesicle that can carry that off somewhere else. Okay, so vesicles, really important to add to your list of organelles that are within the cells. Okay, next one, Golgi apparatus. I'd be totally understanding of you if you wanted to take a break, because there's a lot of information I'm throwing at you. Normally when I teach this, I do it over 2 full lessons because we go through it. I give students time to write it down, to draw it. So I am just throwing mountains of information at you now. Sorry about that, but it's the most efficient way to get it across to you in videos. If you want to pause it, take a break, get a cup of tea, and then we'll carry on, do so. But Golgi apparatus, these are really, really important. They look a little bit like rough or smooth endoplasmic reticulum in that they're still made of these big cisternae, they're still just made of membrane, but they've always got this fairly recognizable structure. There are lots of kind of discs of cisternae piled on top of each other, and they quite often, as you can see with the bottom ones, they start to curve up a bit. Now Golgi apparatus are really, really important because they're the site of protein folding, processing and packaging. So vesicles from the rough endoplasmic reticulum containing the polypeptides that we just talked about will come to the Golgi apparatus. The Golgi apparatus will then do its job of doing the folding and processing, and then it will send out the fully formed products in vesicles out the other side. Okay, so they're a really important kind of finishing school for proteins, but they're that midway point of vesicles that have come from the rough endoplasmic reticulum, get processed back into vesicles to float off somewhere else. So again, illustrating how important vesicles are for transporting things around the cells. Now you do need to know an example of the vesicles that come off from the Golgi apparatus, and the one that always seems to come up in almost all exam boards is a lysosome. Now this is only one example of a vesicle that would come from the Golgi apparatus containing fully formed proteins, and they're named for the proteins that they contain. So in the case of lysosomes, they contain digestive enzymes which are lytic, perform lysis, they cut things, hence lysosome, because it contains lytic digestive enzymes. That's just an example that you could be asked to name in the exam, so make sure you've got that one down. There's only a couple more to go, don't worry too much. Microtubules. Now, microtubules are involved in cell division. I'm hoping this will link up to something that you learnt at GCSE, but in animal cells, microtubules come in specific patterns in structures called centrioles. Centrioles might ring a bell from your GCSEs. When I say specific patterns, if you look at this, the microtubules are arranged in rows of 3, and the rows of 3 all set themselves off around in a circle to make a tube of microtubules. So they look like this one. Now in animal cells, they always come in pairs. There's 2 of them and they're at right angles to each other. So one will be that way, one will be that way. And you find them just outside the nucleus. Now the really important thing that they do is that they produce spindle fibers which are used to pull the chromosomes apart during cell division. We talked earlier in this presentation about single and double chromosomes. Once you've got the double chromosomes, during mitosis, it's microtubules shooting out spindle fibers that grab onto the 2 halves and pull them back into 2 single chromosomes and to separate them so each end of the cell gets a full set of chromosomes. They do play a really, really important role. In plant cells, the microtubules are less well structured than these sort of very regular centriole structure, but plant cells and animal cells both do have these microtubules. And as I've just said, they form the spindle fibers which pull on the chromosomes during cell division. So take a screenshot of this one, but we're going to start putting it all together in a minute. So that's it for the organelles that I'm going to be showing you and teaching you all the little details. But we do need to put it all together. So what we're going to do it for an animal cell, then we're going to do it for a plant cell, and then I'm just going to put up a couple of blanks for you can use to practice on your own if you want to. So if we're looking at an animal cell, we're going to have the cell membrane, you're going to have the cytoplasm, we're going to have the nucleus. But now remember all your labels for the nucleus. You've got to label the chromosomes of DNA. You've got to label the nuclear envelope and the nuclear plasm and the nuclear pores. You've also got that nucleolus structure. So there's a lot of labels to put there just for the nucleus. Also notice how I've labeled the nucleus. I've used a bracket label. The reason for that is the nucleus is a large thing with lots of things to label within it. So it would be foolish to just put one line going to the edge of the nucleus to label it. It would be like putting an arrow directly to my nose and labeling it Ben. Well, no, that's nose, isn't it? Ben is this entire person here. So you want to try and use a bracket label when you're labeling a whole big structure. It's just more clear. Okay, let's add something else on here now. We can put the centrioles just outside the nucleus, as I said. You may want to just label those as microtubules. If this was a plant cell, you would definitely only label them as microtubules. reticulum. The rough endoplasmic reticulum covered in its ribosomes. Notice its location. It's right outside the nucleus, and it always is. That's to minimize the distance that messenger molecule needs to travel. That messenger molecule being the kind of photocopy of the DNA that needs to come out through a nuclear pore and to the ribosomes. So that's where you'd find rough endoplasmic reticulum. You can also get free ribosomes. Not all ribosomes are stuck on rough endoplasmic reticulum. So if you're drawing this, or if you just see tiny little things floating around, it could well be that they're just free ribosomes that aren't associated with rough endoplasmic reticulum. We'll put some smooth endoplasmic reticulum there. I'm not bothering to label the cisternae on all of these things because you've got them on the rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus. I'll be labeling the same thing. You may choose to label the cisternae, that's the different parts of them. I've chosen not to. I'm going to put my Golgi apparatus over there. And of course, because we've got Golgi apparatus and rough and smooth endoplasmic reticulum, I'm going to label vesicles. I would suggest you maybe if you're doing this in the exam, put vesicles, e.g. a lysosome, just because it covers all bases there. I'm also going to add on here a mitochondria. It's a bit more faint, but it's just here. Within the mitochondria, again, a bracket label. You've got the outer membrane, you've got the inner membrane folded into the cristae, and you've got the matrix as the fluid within there. You've also, of course, got a small temporary vacuole. Now, as well as saying the small temporary vacuole, one extra label with the vacuole is that the membrane of the vacuole is called the tonoplast. That's just one extra word, as if you didn't have enough. So there's a huge amount of labels on that screen. So take a screenshot of that because that's really, really useful to use. What we'll do now is we'll switch to a plant cell just to add some more details to this. So take a screenshot of that if you want. Okay, looking at a plant cell then, if we put the cellulose cell wall around it now, then remember the membrane is different because the membrane is now pressed tightly against the cellulose cell wall, but the label looks still in the same place labeling the cell membrane, but we've got cellulose cell wall as an additional thing. We can also put the chloroplasts in, a bracket label again for chloroplasts, and I can add the labels to the chloroplast envelope. I'm not going to worry about labeling the inner and outer membrane. I didn't for the nucleus either, because we can just call it an envelope. I did for the mitochondria, because we generally don't call that an envelope. The liquid inside, of course, is the stroma. I'm not going to label a starch grain because I don't always have a starch grain. So that's pretty much all I need for the chloroplast. So all really that remains for me to do is to change my vacuole from the small temporary vacuole to the large permanent vacuole. You see I've changed the label as well, and that's now everything you need for a plant cell. So take a screenshot of that one as well, so you've got that for your notes. Okay, what I'm going to do now, I'm just going to jump forward to my next slides. So all I've done here on these last two slides, I've taken all the labels off and I've just given you a totally blank animal cell with the labels there. If you want to screenshot this, stick it on a Word document, print it out, you can then practice labeling everything. And I've done the same as well for a plant cell. There you go. Still with all the labels, with a comprehensive list of all the organelles and sub-organelle parts that you need to know. So use those for practice if you need to. Okay, that's everything that we need to cover on cell ultrastructure. Huge amount of information, lots and lots of terms, lots and lots to memorize, but you really do need to know that. From here, I'd like you to go to prokaryotic ultrastructure, just so we can do exactly the same treatment with prokaryotes as we've just done with those two main eukaryotic cells, but there's far less for prokaryotes, so that's a very, very quick video. If you find this useful, please do remember to give it a like, subscribe to my YouTube channel if you haven't done so already, and please do follow me on Instagram and Facebook for lots of additional biological material. Thank you.